a nematologist at Stanford, and he took a look at some of the pictures, and basically there are like 15,000 species of nematode roundworms, and they all look alike, pretty much. So he said PCR is the way to identify them. So that's what we went to. So that's just the same in case the movie didn't work. And so one outside, one leatherback, and 2 slugs had very high infection levels by nematodes in this study. And the greatest number of larvae were, again, from the bottom of the 1502. And Kate followed this out to 20 days, and so larvae were shed from mollusks up to day 11, and then they started dropping off. So for 11 days, that mollusk can be shedding rat lungworm in your tank. And the 5 ml sample she pulled on day 1 initially contained only several nematodes, but then 4 days later, 4 to 5 days later, in the semi-slug, we had this huge peak of those 900. And with the leatherback, it occurred about day 7, and that was very variable, the number she was seeing. And then, of course, there's a lot of error, human error, and it's very hard to see counting 900 larvae. Okay, again, leatherbacks may require more maceration to release large numbers, but the semi-slug certainly does not. And the worms we were looking at were various sizes and shapes. So we wanted to see, one of the other questions is, because in Trish's CTAR manual, she conferred with the CDC, and the CDC said 20 micron filter should be sufficient. I don't think there's any data to back that up. I don't know, Rob, do you know? I've never seen a study, and there's nothing on the CDC website. Trish said, maybe we should go with a 15 micron filter, but they said no, 20, so that's what is out there right now. So you need 20 should be sufficient. Well, an Ash in 1970 measured 35 L3 stage larvae, and his maximum width, this is the diameter 26 microns, but it ranged from 23 to 34. To me, 23, this is variable, okay? 20 is not enough, in my opinion. That's too close.
You said you had a 5.
I have a 5.
I don't doubt it.
And we do drink our water. Okay, Mark Harris and Sanders, who published the study on the life cycle of these guys, are saying that the L1s, their width is about 15 microns, so 15 L1s can go through. And then you jump up to L2s, and L3s are about the same, though their paper says L3s tend to be a little bit thinner than L2s. So is a 20 micron filter sufficient for a catchment system? That's the question. And so, Kate, we bought a couple of these 20 micron metal sieves. Now, this is not the same as a filter you put in your catchment tank, but we just wanted to see, would these guys pass through a 20 micron metal sieve? So again, we actually did several trials of this. First one, she had 2 Martensi and 2 Altae, and the second trial, she did 3 leatherbacks and a few Martensi based on what we could collect from the wild. Tested them by qPCR, and they were positive. And so she continued with the study. The only difference with this one, they were compared similarly to the other studies we just talked about, where they plopped in a 15 ml thing of rainwater in the tube. We wanted to rinse off the outside, so we pre-rinsed these guys to get any external parasites off, and just to try to help figure out what are those worms. Okay, and again, she did 3 samples drawn from the bottom of the falcon tube each day, and she put these into a petri dish with other rainwater that was tested to be negative, and looked at it under the scope. And then she took the sieve and put it into a petri dish and immersed the mesh so that the water, so that the mesh itself was immersed with rainwater. And then she placed the larva on top of the sieve, and then we took the water from below the sieve and tested it. And so the results are that that the samples were positive for an angio. And so she ran these out, so she put the larva through the sieve, let it sit, well, sometimes it was 4 hours, sometimes it was overnight, and then tested the water underneath and kept that water. And she kept that water and looked at it every few days, okay? And she terminated trial 1 at day 53. And then she did this again and terminated it at day 56. Okay, and we tested the water by qPCR, and it was positive. And so just for those of you that are familiar with it, this is a replicate and this is a quadruplicate. These are amplification plots that are sitting right in the center of our references, which is exactly what you want. So these are both clearly, in my mind, clearly positive. And so what that means is that Angiostrongylus can get across a 20 micron sieve. Now that's very different than what a catchment might be. And again, these are the worms she was looking at under the scope. There's a huge amount of them. And they were very much alive after 53 days, after 56 days. This is about 53 days. Okay, so based on this, you know, we can say that these worms can live a lot longer in rainwater. And again, as long as the nutrients are there, they can live for long periods of time. So overall, drowned mollusks can shed viable Cantonensis in rainwater. Most larvae are found in the bottom of the water column. So what we did with our catchment, that's why catchment cells, the stuff you can actually move the intake so that it's floating like a foot below the surface instead of drawing from the bottom, hoping that might help. Viable L3s can be released as a larva, L3 larva swimming, or in a coiled form. And she found that larvae were shed up to day 11, viable larvae. And the peak larvae yields, the days of peak larvae yields are different between species. And viable Cantonensis were able to pass through a 20 micron sieve and live for at least 56 days. And this is where my catchment studies stopped, because we had funding from the UH Research Council to run these trials last summer, and that money, of course, is gone. What we need to do, and what we're collaborating with Hawaiian Catchment is, because they're going to provide us with a little mini catchment system, so we can test lots of different filters, 20 micron, 15 micron, 10, 5, in the laboratory. I have space, empty lab, I can actually put it in. That's a big thing. The problem is I don't have any funding. So I applied to Sea Grant, and even though both reviewers said it was a very good proposal, one of the reviewers said, well, you haven't demonstrated that there's a problem. Well, that's what we do the study for, and also that it wasn't a wide enough range of audience for the Sea Grant mission. So that one is dead in the water, and I don't have any other grants out on this catchment system study at this point in time. So that's as far as we're able to get. Do I have another couple of minutes? Yeah. Okay. So this is one study that we've been working on, and again, I just want to point out that my lab has kind of been taken over by rat lungworm in the last couple of years. We're conducting studies to find out where the rat lungworm hotspots are on the Big Island. Rob's group did a very nice survey of the statewide. We want to focus in on the Big Island, especially the epicenter in Puna. I want to see what, we're switching intermediate hosts. It used to be Cubans, now we have it's Martensi, the monitoring that over time. People always ask me, what should I wash my veggies in? And I can't tell you what to wash them in. Really good potable water, leaf by leaf. We have done some studies. People have used hydrogen peroxide or vinegar. These do not work. No. Our stomach, they live in our stomach acid, so any acids, they love it. And in fact, hydrogen peroxide was what Silka was using, thinking that she was safe, and she had a very severe disease. Possibility of rat lungworm in water, we just talked about that. And diagnostics, there are no diagnostics in the state of Hawaii. And one of the things I did 2 studies last year, both in wild rats working with USDA APHIS and Hilo. And we wanted to see if rat lungworm DNA can be detected in blood. And so we ran a trial, infected rats, low dose and high dose and control, and we were able to demonstrate that yes, we can detect rat lungworm DNA in the blood of rats at certain time points. So this summer I'm going to try that in humans as well. So this paper just got accepted at PLOS 1, so that'll be coming out. To determine the efficacy of the vaccine, and I should say here, we're also optimizing immunodiagnostic methods and working with a lady from Thailand. Her name is Manoa, and she's at the medical center in Bangkok, and she's developed a really nice little test strip where she has an antigen on the test strip. That's what they're using for diagnostics, to test for the presence of antibodies. And so I'm trying to get that up and running. Working with a colleague in Spain, Antonio Osuna, and we tested his vaccine against Cantonensis, which is a related organism, the Cantonensis, and it doesn't seem to work, unfortunately. And then right now I'm conducting this pilot study to look at prevalence in humans for rat lungworm from people, 500 volunteers on the east side of Hawaii. So that study is underway right now. Last but certainly not least is education. This is a very preventable disease, and a large focus of our group is on prevention. And so we developed a curriculum that involves a 2nd grade level book about the mystery of rat lungworm disease. If anybody wants one, just shoot me an email, and I'm glad to have to send you some. The teachers love when we piloted it in 4 schools, and College of Pharmacy paid to have 10,000 of these printed. We have distributed 8,000, and I think it's helping. You educate the keiki go home and educate their families. We also developed a brochure. So we're trying to educate people because it is a very preventable disease if you know about it. So just make sure you wash your dishes. And I'll be glad to take any questions.
Question is my lack of knowledge, is this detrimental to the rat? Will it eventually kill the rat?
No, well, it's probably if you have enough. When we were running our rat trials, we pulled 57 worms out of the heart and lungs of 1 rat. And through the course of these trials, which means 57 worms had to go through their brain, because that's part of the life cycle. We didn't see any odd behaviors in these rats. I don't know how that can, I don't know how that can happen, that they don't have some sort of inflammation, but in the rat it doesn't seem to. I'm sure if in the literature there are maximum amounts of worms. Something's regulating how many worms actually develop in the rat. I don't know what that is.
Next question. What about fowls, chickens, ducks that are eating these
Chickens
things
don't
like
seem
mad?
to get it, yeah. So the big ones on the island are dogs, horses, and mules. There are some bird species that can get it, like parrots, I think, and a couple of others. But chickens, no. Ducks, no. Cats, no. Pigs, no. Horses, yes. And I don't know, I don't think cows.
Yeah,
Frogs.
Frogs can get it. I haven't tested any cold. Have you tested any cold? Need to do that.
A lot of them around.
Yeah.
Thank you for your really fascinating presentation. I was just wondering, can boiling the water help?
If you cook. Yeah, so what we say in here to the kids is check, look at it, clean it, and cook it. Cooking will kill them. Freezing will kill them. The only solutions we've found so far, and on the back of this we have a recipe for what we call a slug jug. So if you have slugs around your house and you step on them, what you're doing is killing the host, the larvae escape and go to another host. But if you put them in a slug jug with a 15% salt solution, and Hawaii sea salt works, it dissolves nicely, just keep a big jug of it and put your slugs using tweezers, tongs or something, gloves. Put the slug in there, the slug drowns, and the larva escape the slug and die. So we recommend a slug jug for capturing. Some people have told me they started washing their vegetables in salt water. That's not really a vegetable wash. Bleach, of course, kills them. Bleach is not a vegetable wash. So right now, the best thing is really check, clean, and cook.
What's the natural occurring incident in fresh water in the, mountain stream?
Oh, the rat lungworm larva?
Yeah.
I don't think anybody knows.
But we can assume it's there.
I assume it's there. Good.
So is there any number, like infected dose, how many, let's say, larvae need to come in?
I don't think anybody, in humans, I don't think anybody really knows.
Like you told, they can survive for 5 days in the, let's say, salt water for 5 days. So like you swim in the ocean.
Well, the ocean is only 3% salt. And apparently, based on the literature, they can live in 3% salt.
Or they can be.
They can live in seawater, yeah. Which is a whole other can of worms.
So there's a risk of swimming then.
Well, you're not likely to run into the, I mean, slugs usually don't go into the ocean. You have to have that slug in that cycle, and it's usually in terrestrial environments.
So you had mentioned that most of them sink to the bottom, and that you have modified your catchment system so that you take the water from a foot from the top.
Mm-hmm.
Do you know if the company that cleans a lot of the systems is working with the other homeowners to try
Yeah,
to do
so
that
that's
same thing as
all
well?
Red Street, that's Hawaii catchment.
Mhm.
You know, there's a couple of catchment companies, they're the main ones I've been working with,
Mhm.
and I know Rex and his wife are very well aware of rat lungworm. And so,
And that's a really good way to,
yeah, and they're the ones that are going to loan us the entire Worthy catchment system to do the testing.
right?
It's not too expensive, right? That's not, that's an issue.
Filter? No, they're not, I mean, not really. I don't remember exactly what they cost, maybe $20. But, again, coming back to education. Some people have been living on catchment and without a filter for their entire lives, trying to make them understand that it's important is a stumbling block.
Looks like the NIH might be interested in it, right?
I'm afraid to go to NIH with this, it's the reviewers. If I just applied to Sea Grant and a Sea Grant person tells me you haven't demonstrated it's a problem.
Because there's a small number of cases.
Well, because they're not familiar with rat lungworm and catchment systems. Going to the NIH panel, they're going to look at rat lungworm data. That's not even a problem. Right? Well, we applied to NSF and that didn't go.
And I've talked to NIH, and they basically say, first of all, they haven't heard of it. And when they do hear of it, I tell them about it, and so it's important in Hawaii. They say, oh, yeah, but is it a problem on the mainland? And it's beginning to be a problem on the mainland, and there's potential with global warming, because it's a tropical, subtropical organism. But basically, they said it's too much of a sort of niche thing. It's not malaria, it's not cancer, it's not AIDS.
And it's only Hawaii.
Yeah.
In Florida, they've had the big outbreak of giant African snails. And you do see stuff in the news once in a while about rat lungworm. And they paid, I don't know how much money to train dogs to go and find these snails so they could get rid of them.
They've already been discovered in other species of snails in Florida.
Oh, yeah. So Florida, Louisiana, and Texas, Yeah. So,
Can I have a last,
wash
can I have a last question? Have you heard of SODIs, the solar water disinfection?
Solar water disinfectant? I
Yeah.
haven't looked at that one. I looked at a number of different chemicals, and the ones that worked had this one chemical, sodium potassium sulfate.
Yeah, SODIS is just UV.
UV, MIT is promoting so easy, they were complete.
I don't know
So
if
I was
UV
wondering
affects
if
these
that works
guys.
for.
I'm gonna throw some of the strata
Well, you mentioned you have a UV at the end of your disinfection
Right, that's
system. Do you
mainly
count
for
it
bags
as
of
being
microbes.
a last line of defense?
Well, mainly for microbes.
Yeah.
I don't know if UV will do anything to these. I know it doesn't do much to C. elegans, but
So you have a last question? You don't say? Okay. So I think it's time to thank her and then. She flew all the way from Hilo this afternoon and then she's going back again right now. So
Thank you very much for your time.
Thank you,
Anybody want
to everyone. Thank you for coming.